Imagine for a second that you are standing at the base of this massive skyscraper.
Okay, I'm picturing it.
But as you get closer, you realize this building, it isn't made of glass or steel or, concrete. It is constructed entirely out of books.
Just a solid stack of books.
Yeah, a solid stack of paperbacks and hardcovers reaching hundreds of stories into the clouds. And your job, like your one and only mission for the rest of your life, is to climb this impossible tower and find a single specific typo.
Just one typo in the whole building.
One wrong letter on one page in one paragraph of one book somewhere on, like, the 73rd floor.
That sounds completely impossible.
Right. And to make it even more interesting, the ink on the pages is heavily smudged from the rain, and some of the pages have been violently torn in half. And the entire text isn't even written in a language you speak. It is written in a code of just 4 letters.
Right, A, C, T, and G.
Exactly.
Yeah.
Billions upon billions of them.
Yeah, that is an incredibly daunting visual, but it really perfectly captures the everyday reality of genomic sequencing. I mean, when a patient walks into a clinic with a mysterious, undiagnosed disease, that skyscraper you just described, that is their genome.
And we're essentially looking for that one typo.
Yes. A genetic mutation, or what we call a variant, that could explain their suffering. I mean, it might literally be the difference between health and disease, or even life and death for you.
And for the longest time, the only way we knew how to find that typo was by throwing complex, handcrafted mathematics at the problem, right? We built these massive statistical engines to read the letters and try to guess where the errors were.
That was the standard, yeah.
But today, we are going on a deep dive into a revolutionary tool that took that entire mathematical playbook and just threw it out the window. It is called DeepVariant.
Right.
And instead of using statistics to read the individual letters of your DNA, it turns the genome into a literal picture. It uses artificial intelligence, specifically image recognition AI, to just look at the DNA and spot the mutations visually.
Which, I mean, initially sounds completely absurd. You don't read a book by stepping back and looking at the page as a painting.
No, you read the words.
Exactly. But as we will explore today, this shift in perspective has fundamentally reshaped the entire landscape of bioinformatics.
So to guide you through this, we have assembled a massive stack of source material. We are talking about the original technical papers from the development teams at Google and Verily, plus clinical benchmarking studies from pediatric hospitals dealing with real patients.
And we also have the developer guides for adding custom data channels to the AI and extensive evaluations across 6 different sequencing hardware platforms.
We're going to synthesize all of this for you. The goal of this deep dive is to explore exactly how this massive paradigm shift happened. We really need to understand why treating DNA as an image works so spectacularly well.
And we'll look at how the AI adapts when the rules of biology suddenly change and what this actually means for the future of medicine.
So whether you are a geneticist trying to catch up on the latest pipeline architectures, or you're just, insanely curious about how deep learning is changing the way we interact with human biology, we are going to break down the how and the why.
And we are going to do it without getting bogged down in impenetrable jargon.
Hopefully.
Ah.
Let's start by looking at the traditional way we have been hunting for these genetic typos. Because to understand why DeepVariant is such a massive leap forward, we really have to understand the sheer weight of the math we used to rely on.
That's a good place to start. So historically, the absolute gold standard for this task, a process the industry calls variant calling, has been a suite of tools like the Genome Analysis Toolkit.
Right. Commonly known as GPA.
Exactly. And these are brilliant, meticulously crafted pieces of software. I mean, they have driven genomic discovery for over a decade, but they rely on incredibly heavy statistical modeling.
Like what kind of modeling?
Well, we are talking about using logistic regression to model the errors in the base readings, hidden Markov models to calculate the likelihood of different read alignments, and naive Bayes classification to ultimately identify the variants.
Okay, let's unpack those terms before we go any further.
Yeah.
Because throwing around things like hidden Markov models is exactly the kind of thing that makes people's eyes glaze over.
Fair enough.
What are these mathematical models actually trying to do when they look at a piece of DNA?
Think of it as a massive probability engine. When a sequencing machine reads a piece of DNA, it isn't perfect. It might read an A, but only be, say, 90% sure it was an A.
Okay.
So logistic regression is a statistical way of trying to draw a dividing line between a true biological mutation and a machine error based on various features, like the quality score the machine gave that specific letter.
Got it. And the hidden Markov model.
A hidden Markov model is a way to handle sequences where you can't actually see the underlying reality, you can only see the output.
Okay, give me an analogy for that.
Imagine you're in a windowless room and you're trying to guess the weather outside, whether it's sunny or rainy, strictly by looking at whether people walking into your building are carrying umbrellas.
Oh, I like that.
Right, so the weather is the hidden state, can't see it. The umbrellas are the observable data. In genomics, the true DNA sequence inside your cells is that hidden state. And the messy, smudged reads from the sequencing machine are the umbrellas.
That makes total sense. And then you mentioned naive Bayes classification.
Yeah, that is the final decision maker. It takes all these probabilities and combines them to basically say, given all this evidence, there is a 99.9% chance this patient has a mutation right here.
But you emphasized the word naive.
I did, yeah. Because naive Bayes relies on a very specific fundamental assumption to make the math computationally possible. It assumes that every piece of evidence is completely independent of every other piece's evidence.
Wait, meaning that if the sequencing machine makes a mistake on one letter, the math assumes that mistake has absolutely no influence on the likelihood of it making a mistake on the very next letter.
That is the core assumption, yes. And mathematically, it makes the equations clean and solvable. But biologically and technologically, it is entirely false.
Because machines don't work in a vacuum, right? If a laser scanner gets slightly miscalibrated while reading a long string of the letter G, that error is absolutely going to bleed over into the next letter. The errors are chained together.
They are highly interdependent. They are influenced by the specific sequence context, like those long runs of the same letter, which we call homopolymers.
Right.
Or they are influenced by the physical chemistry of the sequencing machine itself. If the enzyme used to copy the DNA stutters, the resulting errors are structurally linked. The true, perfect mathematical equation that could model all these complex, overlapping sequential errors across dozens of DNA reads simultaneously.
This doesn't exist, does it?
It doesn't. Human engineers simply cannot write an equation complex enough to capture the messy reality of the physical chemistry.
So for years, the entire field of genomics have been relying on math that we secretly know is slightly wrong, just because it was the absolute best we could do with human brains.
Exactly. It was a necessary compromise. But this is exactly where the DeepVariant breakthrough happens. The developers at Google looked at this problem and realized that trying to write a better, more complex mathematical equation was a dead end.
So what did they do?
Instead, they reached for a tool that doesn't need to be spoon-fed the rules. They used an Inception V2 convolutional neural network.
A CNN. Now, anyone who follows the tech world knows that acronym. That is the exact same family of neural network architecture that revolutionized computer vision.
Yes, exactly.
It's a technology that allows your smartphone to search your photo library for, dogs at the beach. It was designed to classify images on the internet.
The leap of imagination required here is truly profound. To look at a massive text file of genomic probabilities and say, let's just treat this like a photograph.
Right.
DeepVariant takes the candidate variants, which are the places where the patient's DNA reads differ from the baseline reference genome, and it converts that pile of text data into a multidimensional image. Technically, in machine learning, it's called a tensor.
I really want to visualize this for the listener. When we say image, how big of a picture are we actually talking about? Are we looking at the entire genome at once?
Not at all. The image is surprisingly small. It is a highly focused snapshot. The tensor is exactly 221 pixels wide and 100 pixels high.
221 by 100, that's tinier than the profile picture on a social media account. It's just a tiny little rectangle.
It is very small, but it is incredibly dense with information. Within that small pixel grid, the software visually encodes the baseline human reference genome across the top, and then it stacks all the patient's individual sequencing reads underneath it.
Yeah.
And because it is processed as an image, the convolutional neural network doesn't look at the letters one by one sequentially, like that Markov model we talked about. It processes the entire visual block simultaneously.
It takes it all in at once.
Right. It looks for patterns, textures, and spatial relationships across all those reads at the exact same time.
But turning DNA into a picture, I have to admit, it feels like we are throwing away the actual biological meaning of the sequence. It feels like, translating a beautiful, emotionally complex Shakespeare sonnet into a barcode. just so a grocery store scanner can read it.
I can see that.
You might understand the pricing, but you lose the poetry, the context, the meaning.
I see where that skepticism comes from, but the barcode analogy misses a crucial element of what the CNN is actually doing. We aren't translating the DNA into an arbitrary, meaningless pattern. We are mapping it spatially.
Facially.
Yeah, the spatial relationships in those 221 by 100 pixels perfectly capture the physical, biological reality of stacked sequencing reads.
Okay, how so?
Well, think about how a human geneticist actually works. When they are unsure about a mutation called by the software, what do? They open a program called a genome browser, and they literally look at a visualization of the reads.
Oh, I've seen those.
Right. The vertical columns show what base is at what position across multiple different strands of DNA. The horizontal rows show a single continuous strand. The image format preserves that exact spatial relationship, which is precisely where the evidence for or against a mutation lives.
So it's less like turning Shakespeare into a barcode and more like giving a master detective a high-definition photograph of a crime scene rather than just handing them a written list of where the furniture was located.
That is a phenomenal way to put it.
Because the visual context, like the blood splatter next to the broken lamp, that spatial relationship matters.
Yes. The convolutional neural network can basically see the digital equivalent of that crime scene. It might notice that a certain type of sequencing error almost always happens right next to a run of 5 Gs, and only when the read is aligned in a reverse direction.
And we didn't have to program that rule.
Exactly. We never explicitly programmed the AI to look for that specific rule.
Yeah.
It just learned it by staring at millions of examples.
And the results from this visual approach were staggering. The source material highlights that DeepVariant entered the FDA's Precision FDA Truth Challenge.
Right, that was a huge deal.
This is a massive blinded competition to find the most accurate software for identifying SNPs, or single nucleotide polymorphisms, those one-letter typos in the DNA.
And DeepVariant won highest SNP performance. Winning a rigorous FDA challenge is a massive validation. But the true triumph of this architecture isn't just winning a single competition on standard human data. The real magic is its generalizability.
Generalizability, meaning how well it adapts to new situations.
Right. With traditional mathematical models, if you change anything about the underlying parameters, the math breaks. If the scientific community updates the reference human genome, say moving from the older GRC H37 build to the newer GRC H38 build, the developers literally have to manually go in and tweak the statistical priors.
That sounds exhausting.
It is. And if you want to use the software on a mouse genome instead of a human genome, you have to rewrite massive chunks of the underlying logic because a mouse has a completely different background mutation rate and genetic structure.
You essentially have to build a new tool for every single new job.
Exactly. But DeepVariant generalized across these different genome builds and even across different species like the mouse without any human engineer manually tuning the algorithm.
Wait, really?
Yeah. The underlying architecture didn't change at all. You just show the neural network a few thousand pictures of mouse DNA, and it learns the new patterns on its own.
And the papers show it goes even deeper than that. It wasn't just generalizing to new species, it generalized to completely different sequencing technologies. The sources list an incredible array of hardware: Illumina, 10x Genomics, Ion AmpliSec, PacBio, and this older, incredibly difficult technology called solid sequencing.
Oh, solid sequencing is a perfect case study for this. It used a radically different chemistry based on color space coding, and it was notoriously error-prone.
Yeah, the papers note a wild statistic about the solid data. Out of the box, before any advanced filtering, the raw candidate variants had a positive predictive value, a PPV, of just 13.9% for SNPs.
That is terribly low.
Right. That means if the machine flagged 100 spots as mutations, 86 of them were complete garbage, just machine noise.
For a traditional statistical model, a 13.9% PPV is a total nightmare. It's almost impossible to write a clean equation to filter out that much noise without accidentally filtering out the real mutations, too. But the Google team just took that incredibly noisy, messy, solid data and fed it straight into DeepVariant. They trained the neural network on those specific, ugly error patterns.
And DeepVariant pushed the PPV up to 99.0%.
That's incredible.
From 13% to 99%. That is bordering on magic.
It isn't magic, though. It is the power of a deep neural network acting as a universal approximator. By presenting all the data together as an image, the network serves as a blank canvas. It learns the complex, interdependent error patterns that humans just couldn't mathematically model. It figures out the true likelihood function entirely on its own.
So the theory is beautiful, the architecture is revolutionary, and winning a sterile-controlled FDA challenge is a great headline. But the real test of any medical tool isn't how it performs in a coding competition or on a carefully curated benchmark dataset, right?
Right. It is how it performs in the messy, high-stakes, chaotic environment of a real hospital with real patients whose lives are literally on the line.
Which brings us to a pivotal clinical study from the source material. Researchers wanted to pit the old king, GATK, against the new challenger, DeepVariant, in a real-world pediatric setting.
Yes. This was a study focused on trio sequencing, so let's establish what the researchers actually did there. They took 80 pediatric patients, children who were suspected of having rare, undiagnosed Mendelian disorders, and they sequenced the child's DNA along with both biological parents.
Hence, trio sequencing. Mother, father, child.
Exactly. Performing this in a pediatric hospital is the ultimate stress test. These families have often been on diagnostic odysseys for years. They are desperate for answers. And analytically, trio sequencing is incredibly powerful because it establishes a biological ground truth. You aren't just looking at an isolated string of DNA. You are looking at the laws of inheritance in action.
Before we get to the accuracy, let's talk about the operational reality of running a hospital lab. Time is money, and time is answers. The study compared GATK version 4.1.2.0 against an early version of DeepVariant, version 0.8.0.
And the execution time was a massive differentiator right away. Processing a single trio, so that's 3 whole genomes, took GATK 2 hours and 30 minutes. DeepVariant finished the exact same task in 1 hour and 30 minutes, saving an hour per family. I mean, when a clinical lab is processing hundreds of samples a week, that translates to days of saved compute time. It means families get their diagnostic reports faster.
Efficiency is vital for scale, absolutely. But in clinical diagnostics, accuracy is paramount. A fast wrong answer is infinitely worse than a slow right answer.
Very true.
Which forces us to ask, in a real-world hospital setting where you don't have a perfectly curated answer key provided by the FDA, how do you actually measure if the software is making mistakes?
You use the biology against itself. The study relied on the Mendelian error rate.
Exactly. The fundamental laws of Mendelian genetics dictate how traits are passed down. If you sequence a mother, a father, and a biological child, the child's genetics are bound by strict rules. If the variant calling software says the child has a specific mutation, and neither the mother nor the father has that mutation, one of two things has happened.
Right.
Either the child has a rare de novo mutation, which is a spontaneous brand new change that happened at conception, which is biologically possible but statistically very rare, or the software just screwed up. It's a false positive.
Yes.
It's essentially a paternity test for data quality. If the math says the child has a trait that chemically, physically couldn't have been inherited from the parents, the math is wrong.
I love that phrasing, a paternity test for data quality. It perfectly encapsulates the metric. In this study, they analyzed 77 biological trios. They actually had to drop 3 families because the sequencing revealed a non-paternity issue, which, happens in clinical testing.
Oh, wow. Yeah.
Across those 77 families, the Mendelian error rate for GATK was 5.25%.
Meaning over 5% of the unique calls in the child were biologically impossible.
And for DeepVariant, the Mendelian error rate was only 3.09%.
That is a significant drop in false positives. The AI hallucinates less often than the statistical model. But they didn't just rely on Mendelian errors. They brought in another metric that requires a bit of a chemistry lesson, I think. The tight-TV ratio.
Right, the transition to transversion ratio. To understand why this is a metric for accuracy, we have to look at the physical shape of DNA.
Okay.
DNA has 4 bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are called purines. If you look at them under a microscope, they have this bulky, double-ring chemical structure.
Okay, purines are double ring.
Cytosine and thymine are pyrimidines. They have a smaller, single-ring structure.
So you have the double ring bulky ones and the single ring skinny ones.
Exactly. A transition is a mutation where a bulky purine swaps with the other bulky purine, so A becomes G or G becomes A. Or a skinny pyrimidine swaps with the other skinny pyrimidine. Because the physical size of the molecule stays roughly the same, the overall shape of the DNA double helix isn't heavily distorted.
But a transversion is when you cross the streams. A bulky purine is swapped for a skinny pyrimidine.
Yes, and that physically distorts the DNA helix. It creates a bulge or pinch in the strand. Because of that physical distortion, the body's natural DNA repair enzymes catch and fix transversions much more easily than transitions. Therefore, in nature, transitions happen much more frequently.
That makes perfect sense.
So for whole exome sequencing, which focuses only on the regions of the DNA that code for proteins, decades of biological research have established that the true biological ratio of transitions to transversions should be right around 3.0.
So if your software spits out a list of mutations and the ratio is way off from 3.0, you know your software is catching a lot of artificial machine noise.
Yeah.
Because random machine errors don't care about the physical geometry of purines and pyrimidines, they just make random mistakes, which drives the ratio down.
Precisely. A higher ratio, moving closer to 3.0, indicates a cleaner data set with more true biological mutations and fewer machine artifacts. In this hospital study, JTK achieved a TIT-TV ratio of 2.04. DeepVariant scored 2.38.
Both are under the perfect 3.0. Because sequencing is always somewhat noisy, but DeepVariant is significantly closer to biological reality. It is providing a cleaner picture.
Absolutely.
But let's get to the metric that actually matters to the patient sitting in the waiting room. Diagnostic yield. Did the software actually find the mutation that was causing the child's disease?
Out of the 80 families in the study, 40 ultimately received a genetic diagnosis.
A 50% success rate, which for anyone outside the field might sound low, but for complex, rare pediatric diseases, that is sadly the standard baseline.
It is. Within those 40 diagnosed families, there were a total of 63 disease-causing variants responsible for the conditions. DeepVariant successfully detected 62 of them. JTK detected 61.
They both performed incredibly well, but as we know in science, the failures are often more instructive than the successes. Let's look at the variant that DeepVariant caught, but JTK missed.
The clinical researchers dug into this specific miss. It turned out the variant was located in an area with very low sequencing coverage. The read depth at that specific spot on the genome was only 4.
Meaning, out of the billions of letters processed, the sequencing machine only managed to capture that specific disease-causing letter 4 times.
Right. JKTK's statistical models looked at a depth of 4, ran the probabilities, and decided it was just too uncertain. It filtered the mutation out as noise. But DeepVariant's image recognition looked at the spatial arrangement of those exact same 4 reads and was confident enough to call it a true mutation. And clinical validation proved DeepVariant was right.
It really highlights how robust the visual AI is, even when it's starved of data. But what about the variant they both completely missed? Out of 63 variants, both pipelines failed to find one of them.
Yeah, both pipelines completely missed a large deletion in a gene called OTC, specifically in exon 2. A 139 base pair chunk of DNA was just missing from the child's genome.
And the authors of the study point out that this failure was entirely expected, right?
Yes, it's a limitation of the specific architecture. Both GALATE-K and this version of DeepVariant are designed and optimized exclusively to find small variants, typically SNPs, or very small insertions and deletions under 50 base pairs. A 139 base pair deletion crosses the threshold into what we call structural variants. It requires an entirely different class of bioinformatic algorithms to detect.
It's like criticizing a high-powered optical microscope for not being able to see a galaxy. It's not a flaw in the microscope, it's simply the wrong tool for that scale of observation.
Exactly. So summarizing the clinical trial, the image-based approach isn't just a neat theoretical trick. In a real hospital, it is faster, it generates fewer biologically impossible errors, and it finds the variants that save lives, even when the data is sparse.
But here is where the story gets deeply fascinating for me. Because the technology landscape of genomics is chaotic. It never sits still. Every few years, a company invents a radically new sequencing machine that produces completely different types of data signals.
It's constantly evolving.
If you build a highly specific statistical model for an Illumina machine, and then a brand new technology launches, your model is obsolete. Does the entire Google AI need to be rebuilt from scratch every time a hardware engineer creates a new laser scanner?
That leads us to a brilliant piece of software engineering from the developers, which they outline in a guide on the custom channel framework. To answer your question, no, you absolutely do not rebuild the neural network. You just give it more colors to look at.
Okay, let's unpack how these tensor images are actually constructed. Earlier, we said the image is a tiny box, 221 by 100 pixels. But it is not a flat black and white photograph. It is incredibly deep.
Right.
A standard digital photo on your phone uses 3 channels to encode data: red, green, and blue. By mixing those 3 channels, you get every color imaginable. But DeepVariant doesn't use RGB. It traditionally uses 6 biological channels.
The documentation explicitly lists these 6 core channels. First, the read base, so what letter the machine thinks it saw. Second, the base quality, which is a mathematical score of how confident the machine is in that specific letter. Third, the mapping quality, so how confident the alignment software is that this tiny fragment of DNA actually belongs in this exact spot on the massive human genome.
Okay, that's 3.
Fourth, the strand of alignment is the read going forward or backward. Fifth, does this specific read support the candidate variant? And sixth, does the base differ from the baseline reference genome?
And every single one of those data points is assigned a numerical value between 0 and 255.
Yeah.
Just like the intensity of a red pixel in a digital photo, they stack these 6 channels on top of each other to create the full tensor.
And the physical layout of the image is structured meticulously. The top 5 pixels form a reference strip, which is a visual representation of the perfect, healthy baseline genome. Everything below that strip is the read pileup, the messy, real-world data from the patient.
But the genius is the flexibility, right? The developer guide, literally titled Adding Custom Channels, explains how any researcher, anywhere in the world, can add new channels to this tensor image. If a new machine produces a weird new signal, you don't rewrite complex statistical Bayesian theorems.
No, not at all.
You just write a tiny script to extract that new signal, map it to a scale of 0 to 255, and slide it into the image as a 7th or 8th color.
The elegance of this framework is profound. You are decoupling the biological interpretation from the complex math. Let me give you an example from the source material. They detail an experiment where they added a custom channel called insert size.
To understand that, we need to know how standard Illumina sequencing works. It's called paired end sequencing. Imagine you want to read a page of a book, but you can only read the very first and the very last sentence. The insert size is the physical distance between those 2 sentences. It's the length of the unread DNA fragment stretching between the 2 ends you actually sequenced.
Knowing that physical distance can provide massive context about structural abnormalities. So the developers just took that distance, assigned it a pixel brightness value from 0 to 255, and added it as a new channel. They basically gave the neural network a new color that represented physical length.
And what happened? Adding just that one single channel made the entire model 4.7% more accurate on whole genome sequencing and a massive 9.8% more accurate on whole exome sequencing, just by letting the AI see the distance between the reads.
They did the same thing with a channel called read orientation, which captures bitwise flags from the alignment files, so data about whether a read is a primary alignment or a supplementary one. And the true test of a framework is whether other people can use it. The sources note that a completely separate company, Genomics, used this framework for their brand new, radically different sequencing platform.
The Ultima platform is fascinating. It uses a flow-based sequencing method that produces very specific types of errors. It struggles with homopolymers, and it outputs custom probability scores for missed chemical signals.
A traditional variant caller would completely choke on that novel data format, but the Ultima engineers just grabbed those unique probability scores, encoded them as custom channels, fed them straight into DeepVariant, and trained the model. The neural network learned the unique quirks of the Ultima hardware simply by looking at the new colors.
Which naturally leads to a question. If adding the insert size channel makes the whole system 5 to 10% better, why stop there? Why limit this image to just 6 or 7 channels in a tiny 221 by 100 pixel box? Why not add 100 channels? Why not map every conceivable metric, temperature gauge, and laser voltage reading from the machine into a massive 4K resolution image and let the AI figure it all out?
It is the eternal temptation in machine learning. More data equals better results, right? But the reality is a constant battle between 3 forces: computational overhead, the noise-to-signal ratio, and the ever-present danger of overfitting.
Let's start with computational overhead. Every channel you add is another matrix of data that has to be stored in RAM, processed by the CPU, and pushed through the GPU during training. If you make the image 10 times larger, processing a single patient's genome might go from taking 1 hour to taking 3 days. That breaks the clinical utility.
But the bigger danger is overfitting. Think of a student studying for an exam. If you give them a short study guide with the core concepts, they learn the rules. If you give them a 10,000-page textbook that includes every piece of trivia ever recorded, they might just try to memorize the textbook word for word instead of learning the underlying concepts.
Right.
When they take the test and the questions are slightly different, they fail.
So if you feed the neural network 100 channels, many of which contain random machine noise, the AI might find a completely artificial pattern. It might decide, every time pixel 47 in channel 82 is dark, it's a mutation. It memorizes the noise instead of learning the biology.
That is overfitting. The 221 by 100 pixel size and the limitation to a core set of highly informative channels was heavily optimized by the developers. It provides just enough local context for the AI to be highly accurate without providing so much noise that the model hallucinates.
So the custom channel framework proves that DeepVariant can adapt to new machines seamlessly. We've established it can learn new hardware. But let's push this architecture to its absolute breaking point. What if the underlying hardware is exactly the same, but the fundamental rules of the biology completely change?
That brings us to one of the most remarkable case studies in the entire stack of sources, the Malaria GN project.
We've been talking about humans and mice, but the Malaria GN researchers took this advanced, human-trained AI and pointed it at the genome of Anopheles gambiae, the species of mosquito that transmits malaria. They were looking at a pedigree, a family trio, just like the pediatric hospital study. They sequenced mother mosquito 802331C, father 802333C, and child 802234C.
They took the standard out-of-the-box DeepVariant model, the one that had won the FDA challenge, the one that had conquered the pediatric hospital, and ran it on the mosquito data. And the result was an absolute catastrophic failure.
Remember the Mendelian error rate, the paternity test that checks for biologically impossible mutations. In the human hospital, DeepVariant had a phenomenal error rate of about 3%.
Yeah.
When they ran that same AI on the mosquito child, the Mendelian error rate skyrocketed to 16.92%.
Nearly 17% of every genetic call the software made in the child was mathematically impossible based on the parent's DNA. It was essentially producing garbage.
The researchers had to figure out why this highly advanced AI was suddenly failing so spectacularly. And when they opened up the data and looked at the specific errors, they found something incredibly strange. DeepVariant was aggressively calling a massive number of sites as homozygous reference.
Homozygous reference is a genetic term basically meaning I am looking at this spot in the DNA and it perfectly matches the baseline genome. There is no mutation here. Everything is normal.
But when the human researchers looked at the raw visual reads for those exact same spots in the mosquito child, the data was screaming with alternate alleles. There are obvious glaring mutations stacked up in the reads. The AI was staring directly at a massive pile of evidence proving a mutation existed, and it was calmly outputting, nope, perfectly normal, no mutation.
Right.
Why on earth would an AI intentionally ignore reality?
To understand this, we have to look at the evolutionary biology of the mosquito versus the human. It comes down to a concept called variant density. Humans are, from an evolutionary standpoint, a relatively young species. We have very long generation times, 20 to 30 years before we reproduce, and we have very few offspring. Because of this, our overall mutation rate is relatively low, and our mutations are spread out. You rarely see multiple true mutations clustered tightly together in a small stretch of human DNA.
But mosquitoes are completely different. They have incredibly short generation times, they reproduce in days, they produce massive numbers of progeny, and they face intense evolutionary pressure from things like insecticides.
Because of that biology, mosquitoes generate and tolerate a vastly higher density of genetic variation. In a mosquito genome, it is entirely normal and biologically real to see 3, 4, or 5 distinct mutations clustered tightly together within a 100 base pair window.
Now, put yourself inside the brain of the human-trained neural network. For its entire existence, it has only ever looked at human DNA. In the human genome, if you suddenly see a dense cluster of 5 mutations right next to each other, it's almost never a real biological event.
In humans, a dense cluster of variants is almost always a mapping artifact. It happens when a piece of DNA from a highly repetitive region of the genome gets accidentally mapped to the wrong location by the alignment software. The reads look like they are full of mutations, but really they just don't belong there.
So, when the human-trained AI was unleashed on the mosquito genome and saw those real clustered mosquito mutations, its human training kicked in. It looked at the dense cluster and said, I've seen this exact pattern before in humans. It's a mapping error. It's just noise. Ignore it. Force the cull to homozygous reference.
Yeah.
The AI wasn't broken. It was making the smartest possible decision based on the universe it was trained in. It literally couldn't believe what it was seeing because its priors were so strongly tuned to the quiet, sparse landscape of human biology.
It's an incredible example of how AI bias works in biology. The model was perfectly executing the rules of a world that no longer applied.
It's like training a highly advanced self-driving car, purely in the flat, dry Arizona desert. The car learns exactly how to drive. Then, you drop that car into the middle of a blizzard in New York City. The car isn't broken. The cameras work, the steering wheel works, the brakes work.
Right.
But the car has never seen a snowflake. It sees the swirling white static on the cameras, assumes the sensors are malfunctioning, and slams on the brakes in the middle of the highway. The developers realized they had to teach the AI the weather of the mosquito genome.
But that creates a massive new problem.
Yeah.
In supervised machine learning, to teach an AI, you need an answer key. You need a massive data set of perfectly labeled true examples. For humans, we have the genome in a bottle, these perfectly sequenced reference genomes we've spent billions of dollars verifying. For this specific family of mosquitoes, there was no gold standard answer key.
So how do you train the AI when you don't know the right answers yourself?
You build what the developers called a silver standard. They used the power of the pedigree, the family tree. They looked at the mother and father mosquitoes. They aggressively filtered the parents' data, looking only at the sites where the calls were incredibly clear, highly confident, and undeniably accurate.
Because if you know with absolute certainty what the mother and father have, the laws of Mendelian genetics dictate exactly what the child must inherit.
They isolated these known, mathematically undeniable transmissions. They essentially created a curated list of sites and said to the AI, look at these specific clustered variants in the child. We know for a fact these are real biological transmissions from the parents. This is not a mapping error. Learn this new pattern. They retrained the neural network layers entirely on this mosquito-specific reality.
And the turnaround was breathtaking. When they ran the retrained mosquito-specific DeepVariant model, the Mendelian error rate plummeted. It dropped from nearly 17% down to 2.8%. And when they looked at the most highly confident calls, the error rate hit an astonishing 0.22%.
It went from a catastrophic failure to vastly outperforming every traditional mathematical tool on the market on the exact same mosquito data.
It proves a fundamental point about deep learning in genomics. The architecture, the idea of turning DNA into an image and using a CNN, is universally sound. The model isn't hard-coded to human biology. It is a blank slate capable of learning the specific quirks, the variant density, and the evolutionary history of any species on Earth, provided you can give it the right curriculum to learn from.
We've seen how it adapts to new machines with custom channels and how it adapts to new species by retraining the weights. But what happens when the actual physical shape of the data undergoes a massive technological revolution? A shift was about to hit the genomics world that would break the 221-pixel model in an entirely different way.
Which brings us to the rise of long-read sequencing and the massive computational hurdle known as the haplotype challenge. Up until this point in the source material, everything we've discussed has been built around short-read sequencing, primarily the technology dominated by Illumina.
Short-read sequencing is an incredible technology. The machines shatter a human genome into millions of tiny fragments, usually about 150 base pairs long. They read those tiny fragments with incredible accuracy, and then complex alignment software pieces them all back together like a massive jigsaw puzzle. It is highly accurate and very cost-effective.
But short reads suffer from a fatal flaw. They lose long-range biological context.
To understand why that matters, we have to look at the physical structure of human chromosomes. You have 23 pairs of chromosomes. For every chromosome, you have one copy inherited from your mother and one copy inherited from your father.
Let's say you are analyzing a specific gene that is critical for heart function, and you find 2 different mutations located in that gene. They are separated by a gap of 1,000 base pairs. Knowing the spatial relationship between those 2 mutations is a matter of life and death. Are both mutations on the same copy of the chromosome, meaning they are physically on the strand you got from your dad? That is called being in cis. Or is one mutation on the dad's copy and the other mutation on the mom's copy? That is called being in trans.
Grouping these linked variants together to determine which parent strand they belong to is a process called phasing. You are phasing the variants into a complete haplotype. If the mutations are in trans, one on each copy, it often means both copies of the gene are broken, leading to severe disease. If they are in cis, both on one copy, you still have one perfectly healthy copy from the other parent, and you might be completely asymptomatic.
The problem with short read sequencing is that the reads are only 150 letters long. They physically cannot stretch across a 1,000 base pair gap. The machine sees the first mutation, it sees the second mutation, but it has absolutely no idea if they are connected. It's like finding two torn corners of a photograph. You know they exist, but you don't know if they came from the same picture.
But a new generation of hardware emerged, long-read technologies pioneered by companies like PacBio and Oxford Nanopore. Instead of shattering the DNA, these machines pull incredibly long, continuous strands of DNA through tiny pores or optical sensors. They produce reads that are 10,000, 20,000, or even 100,000 base pairs long.
These massive reads easily cover those large gaps. They can see both mutations simultaneously on the exact same physical piece of DNA. They provide the ultimate long-range context. They solve the phasing problem.
But they created a massive problem for DeepVariant. Remember the image tensor? The developers spent years optimizing it to be exactly 221 pixels wide. That window was perfect for short reads. But when you feed a 20,000 base pair long read into a 221 pixel window, the AI is wearing blinders.
It just can't see it.
Right. It cannot see the long-range context that the new machines are providing. It was blind to the haplotype.
So how do you provide massive 1,000 base pair context to an AI that stubbornly only looks through a tiny 221 pixel window? The initial solution, which Google rolled out in DeepVariant version 1.1, was what we can only describe as a computational Rube Goldberg machine. It was a three-step process.
Step 1, you take your massive long-read dataset and run it through the standard DeepVariant model. The model finds all heterozygous sites. These are the locations where the genome has one mutant copy and one normal copy, but it doesn't know which parent they belong to.
Okay, step 2. You take that list of heterozygous sites, export it out of DeepVariant, and feed it into a completely separate third-party piece of software called WhatsApp. WhatsApp is a phasing algorithm. It looks at the long reads spanning those heterozygous sites, does the complex math to figure out the phasing, and then physically alters the data files. It haplotags the reads.
It's like attaching a digital sticky note to every single read. HP equals 1 means this read came from the maternal haplotype. HP equals 2 means it came from the paternal haplotype. And HP equals 0 means the read was too ambiguous to tell.
Step 3. You take this newly tagged data, load it back into DeepVariant, and run the entire genome again. But this time, DeepVariant activates a new custom channel, the haplotype channel.
And this is where the visual genius of the AI shines again. Instead of just dumping all the sequencing reads into the image tensor randomly, it sorts them spatially based on the sticky notes. It physically segregates the data in the image. It puts all the ambiguous HP equals 0 reads at the top of the pileup. It puts all the maternal HP equals 1 reads in the middle block. And it puts all the paternal HP equals 2 reads in a solid block at the bottom.
By physically segregating the reads into distinct visual blocks inside the image, the neural network could suddenly see the haplotype context without needing the window to be any wider. It just looks at the vertical blocks.
The benchmarking in the papers shows this visual sorting a massively improved accuracy, specifically for INDLs, those insertions and deletions. The sources give a fantastic example regarding homopolymer regions, which are the Achilles heel of long-read machines.
Long-read machines, especially the early ones, struggle to accurately count long, repetitive strings of the same letter. If there is a string of T's, the machine might read 7 T's, then 8 T's, then 6 T's, then 7 T's.
So let's say the patient is at the doctor. Did the patient inherit a true biological sequence of 7 T's from their mom and a true mutation of 8 T's from their dad? Or is the patient actually 7 T's on both sides and the sequencer just randomly stuttered and added an extra T to a bunch of random reads?
When all the reads are mixed randomly in the image tensor, it just looks like a blurry, confusing mess of 7s and 8s. The AI struggles to tell the difference between true inheritance and machine noise, but when you segregate the reads by haplotype,
the visual truth emerges.
Yes. The AI looks at the HP equals 1 block, the mom's reads, and sees it as a solid, consistent block of 7 T's. It looks down at the HP equals 2 block, the dad's reads, and sees a solid, consistent block of 8 T's. By physically separating the biological sources, the noise vanishes. The AI easily calls it as a true inherited mutation.
Knowing exactly which parent the read came from is the ultimate filter against machine artifacts.
So this three-step WhatsApp process worked beautifully. It proved the concept. But as anyone who has worked in computational pipelines knows, a three-step process involving exporting data to third-party software and rerunning the entire analysis is a logistical nightmare.
Oh,
It
absolutely.
was incredibly clunky, required massive storage space, and doubled the processing time. As new long-read machines flooded the market, the developers knew they needed a universal, elegant solution.
They needed to bring the phasing inside the house. The DeepVariant team decided to abandon the external WhatsApp software and build an internal, approximate local haplotagging engine directly into DeepVariant's data preparation phase.
And the way they achieved this is a masterclass in computer science. They didn't use deep learning to solve the phasing. They used a classic algorithmic technique called dynamic programming.
Dynamic programming is a method for solving incredibly complex optimization problems by breaking them down into a sequence of simpler, overlapping subproblems.
Imagine you're trying to drive across the country, and you want to find the absolute cheapest route based on gas prices. You could try to calculate every single possible route simultaneously, which would melt your computer. Or, using dynamic programming, you start at your destination and work backward, calculating the cheapest step to the previous gas station, saving that answer, and slowly building the optimal path back to your driveway.
In genomics, they apply this to the DNA. The internal engine processes the human genome in chunks of 25 kilobases. For each chunk, it builds a massive mathematical graph in the computer's memory. The vertices, the points on the graph, represent the specific alleles, the actual genetic variations observed in the data. The edges, the lines connecting those points, represent the physical long reads that overlap those variants.
The algorithm traverses this massive graph, calculating the mathematical cost of assigning every read to either haplotype 1 or haplotype 2. It scores the best possible phase assignments by working its way to the end of the 25 kilobase chunk and then backtracking along the optimal path to finalize the assignments.
It does all of this complex mathematical graph theory on the fly, entirely in the system's memory, milliseconds before it generates the image tensor for the neural network.
I have to pause here to point out the beautiful irony of this entire system. We started this deep dive by talking about how Google revolutionized the field by throwing away complex, handcrafted mathematical models. They replaced logistic regression and Markov models with a neural network that just looks at pictures.
Right.
But to make that AI faster and more accurate interpreting long reads, what did the engineers have to do? They had to build a highly complex mathematical graph, dynamic programming, back into the very foundation of the data pipeline.
It is deeply ironic, but it highlights a profound philosophical truth about the current state of computational biology. We are not experiencing a war between traditional computer science algorithms and deep learning AI. We are experiencing a symbiosis. You use precise deterministic computer science algorithms, like dynamic programming, to parse and organize the raw data in a biologically meaningful way. And then you use deep learning to interpret the noisy, fuzzy visual reality of that organized data.
And the performance of this hybrid internal haplotagging system is stellar. The benchmarking data proves that the accuracy is virtually identical to the clunky, slow, what's hap method. DeepVariant's dynamic programming hit 99.14% haplotagging accuracy, compared to WhatsApp's 99.26%.
It sacrificed less than 0.1% of accuracy to achieve massive gains in speed and computational efficiency.
By building it internally, they created a truly universal variant caller. DeepVariant can now ingest almost any long-read data format natively, process the phasing in memory, and generate the tensors without any external dependencies.
Let's look at the results when they unleash this universal model on the absolute bleeding-edge sequencing hardware. The PacBio Revio is a massive, high-throughput, long-read machine. It pushes out reads that are 15,000 to 20,000 base pairs long, and it can generate 30x coverage of a complete human genome incredibly fast.
In DeepVariant, utilizing the internal dynamic programming, achieved a staggering S&P F1 score of 0.9993 on the Revio data. That is effectively near-perfect accuracy on human single nucleotide variants.
It also handled the newest, most complex chemistry from Oxford Nanopore Technologies, specifically their R10.4 chemistry. This platform is wild because it produces two entirely different types of data in the same run. It produces simplex data, where the machine reads one strand of the DNA molecule. Simplex data has about Q20 quality, meaning it is 99% accurate, but the reads can be over 100,000 base pairs long.
And it also produces the new duplex data. In duplex mode, the nanopore physically pulls the forward strand of the DNA through the sensor and then immediately pulls the reverse strand of that exact same molecule through the sensor. By reading both sides of the physical molecule, the quality jumps to Q30, 99.9% accurate, with reads typically between 10,000 and 50,000 base pairs.
Nanopore data, because it relies on changes in electrical current rather than optical pictures, has historically been notoriously difficult for variant callers. The error profiles, especially around those tricky homopolymers, are entirely unique.
But DeepVariant handled both the simplex and the duplex data natively. For the ONT duplex data, the combination of custom channels, internal haplotagging, and the CNN achieved the highest variant calling accuracy ever reported for nanopore long reads, a S&P F1 score of 0.999 and an Andel F1 score of 0.90.
It is a monumental engineering achievement. So we have all these different machines, Illumina short reads, PacBio Reveal long reads, Nanopore simplex and duplex. We have different sample preparation methods. We have dozens of variant calling software options. For a clinical lab director or research scientist, how do you actually know what combination works best in the real world? How do you cut through the marketing hype of these tech companies?
You need an ultimate unbiased showdown, a massive benchmarking gold standard, which brings us to the final major study in our stack of sources.
This was a comprehensive study that benchmarked all these variables using the Genome in a Bottle, or GIAB, truth sets.
The Genome in a Bottle Consortium provides the universally accepted reference genomes. They are the undeniable answer keys that the entire industry uses to test accuracy.
The researchers threw everything at these GIB genomes. They looked at multiple generations of Illumina instruments, the Novasec, the HiSec X, and the HiSec 4000. They included the PacBio Sequel 2 Secker for long reads. They compared whole genome sequencing, which looks at everything, against whole exome sequencing, which only looks at the 1 to 2% of the genome that actually builds proteins. And crucially, they looked at different chemical preparation methods in the lab, specifically PCR-free versus PCR-positive.
Let's start right there, because lab chemistry heavily dictates what the AI ultimately sees.
Yep.
What is PCR and why does it matter?
PCR stands for polymerase chain reaction. It is a Nobel Prize-winning chemical process used to amplify DNA. When you extract DNA from a patient's blood or saliva, you often don't have enough physical material for the laser scanners to read accurately. PCR uses enzymes and temperature cycles to basically photocopy the DNA, turning a few molecules into millions of identical copies. A PCR-positive preparation means this amplification step was used before sequencing. A PCR-free preparation skips it, which requires a much larger initial blood sample but avoids the chemical manipulation.
The benchmarking study revealed a critical finding. PCR-positive whole genome preparations have lower overall accuracy than PCR-free preparations. Specifically, the data showed they deeply struggle with calling INDLs, those insertions and deletions. Why does photocopying the DNA introduce errors?
Because the biological photocopier isn't perfect. The polymerase enzyme used in PCR is literally a tiny molecular machine walking down the DNA strand, adding matching letters. When it hits a repetitive sequence, like our favorite example, a homopolymer run of 10 A's, the enzyme can physically slip. It momentarily detaches, loses its place, and reattaches. In the process, it might accidentally add an 11th A or skip one and only copy 9.
It introduces an artificial insertion or deletion into the physical DNA in the test tube before the sequencing machine ever even touches it.
Exactly. The sequencer reads the slipped DNA perfectly, and the variant caller looks at the data and says, there's an insertion here. Distinguishing a chemical PCR slip from a true biological mutation inherited from a parent is incredibly difficult for standard statistical tools.
But the benchmarking study notes that DeepVariant handles these PCR-introduced errors significantly better than traditional tools like GATK.
And the reason why goes back to the beauty of the neural network. The Google developers explicitly included massive amounts of PCR-positive data in DeepVariant's training sets. When the CNN looks at the 221 by 100 pixel image, it has learned exactly what the subtle visual signature of a PCR slip looks like in the read pileup. It recognizes the texture of the chemical artifact and actively learns to filter it out, preserving the true biological signal.
The study also uncovered some fascinating hidden quirks about the specific sequencing instruments themselves. For example, they found that runs on the newer, faster Illumina Novasec actually had slightly lower SNP accuracy, but higher INDL accuracy compared to the older HiSec X machines.
And even more critically, the Novasec data showed distinct, dramatic drops in sequencing coverage in specific genomic regions that had very low GC content.
Low GC content, meaning regions of the human genome where the sequence is mostly made up of adenine and thymine, less than 25% guanine and cytosine. Why does the laser scanner care what letters it's looking at?
Because G and C bases share 3 hydrogen bonds, while A and T only share 2. GC-rich DNA is physically tighter and melts at a different temperature. Different sequencing platforms use slightly different physical chemistries, and the Novosex chemistry evidently struggles to efficiently process regions heavily biased toward A and T.
This highlights the terrifying hidden technical variables that impact medical research. If a scientist is studying a rare genetic disease and the causative mutation happens to be located in a low GC region of the genome, and they decide to sequence their patients on a Novosec to save money, they might completely miss the mutation simply because the physical chemistry of the machine didn't cover that area well.
It emphasizes why researchers must deeply understand the interaction between the biology, the hardware, and the software. It's an ecosystem.
Let's shift to the findings regarding the exome. Whole exome sequencing uses chemical baits or molecular probes to essentially go fishing in the genome, pulling out only the vital protein coding regions and washing the rest away. The benchmarking study compared 3 of the most popular exome capture kits on the market: Agilent, IDT, and TrueSec, and they found massive variability between them.
Huge differences in capture efficiency. The scientific community often treats all exome sequencing as equivalent, but the choice of the chemical kit significantly impacts what regions of the genome you can actually see.
The data showed that the Agilent kit captured a much broader, wider area of the exome. It pulled down more DNA overall, but the IDT kit had significantly higher accuracy on the core target regions that all the kits shared.
Think of picking an exome capture kit, like choosing a fishing net for a research expedition. The Agilent net is incredibly wide. You cast it into the genomic ocean, and it catches a massive amount of material. You might discover something new on the fringes of the exome, but the wide mesh means you also pull up a lot of bycatch off-target reads that dilute your sequencing power. The IDT net, on the other hand, is smaller but much more precise. It catches exactly the target fish you were aiming for with high efficiency, but you cover far less of the ocean.
If you are hunting for a known specific clinical mutation, you want the precision net. If you're doing exploratory research, you might want the wide net. But the final and perhaps most impactful finding from this massive benchmarking study was the direct comparison of short reads to long reads regarding what researchers call the ceiling of accuracy.
The researchers mapped the variant calling accuracy of the software against the depth of coverage, meaning how many times the genome was essentially photocopied and read. For short-read Illumina data, the graph revealed a stark reality. Simply throwing more reads at the problem has massive diminishing returns.
If you sequence a genome at 30x coverage, you get a certain accuracy. But if you double your budget and sequence it at 60x coverage, the accuracy barely improves.
Because you hit the inherent physical limitations of the short-read technology. More 150 base pair reads do not help you phase distant variants, and more short reads do not help you map right through the middle of a massive 10,000 base pair repetitive region. You are just stacking more blindfolds on top of each other.
But the PacBio Hi-Fi long reads told a completely different story. The data showed that even at a significantly lower coverage, just 20x, the PacBio Hi-Fi had fewer non-callable exons and missed genes than the Novosec did at 40x coverage. The sheer length of the reads drastically reduced S&P false negatives because of their vastly superior mapping ability. They don't get lost in the repetitive regions.
And crucially, as the researchers mathematically increased the coverage of the PacBio data, the errors continued to fall. The curve didn't flatten out like the short reads. Long-read technology, paired with DeepVariant's internal haplotagging, has a much higher ultimate ceiling for accuracy. As the technology scales and the per-genome costs inevitably come down, long reads are poised to become the definitive, undeniable standard for resolving the most complex, difficult regions of human biology.
Which brings us to the end of this incredibly dense, fascinating deep dive. We have covered a staggering amount of technical ground today.
We truly have. We traced the entire evolutionary arc of bioinformatics over the last decade. We watched the industry hit the absolute limitations of independent statistical mathematical models. We witnessed the incredible paradigm shift to the flexibility of image-based deep learning, realizing that you can use a convolutional neural network, software originally designed to classify pictures of cats on the internet, to visually read the fundamental language of life.
We saw how that AI conquered the messy, emotional clinical realities of pediatric trio sequencing in hospitals. We watched it stumble and then rapidly adapt to the extreme variant density of the mosquito genome. We unpacked the elegance of the custom channel framework, and we explored how developers fused the classical computer science of dynamic programming with deep learning to phase massive long reads on the fly. DeepVariant has truly democratized high-accuracy variant calling across different hardware, different chemical setups, and completely different species.
It stands as a profound testament to how creative, lateral software engineering can completely shatter the perceived boundaries of biological discovery.
Before we wrap up, let's bring this massive technical journey back down to earth, to why this actually matters to your health. Genomic medicine is no longer science fiction. It is rapidly becoming the standard of care. If you or your child or your parent ever needs to get a genome sequence to diagnose a baffling rare disease or to tailor a highly specific targeted cancer treatment, it is overwhelmingly likely that an AI looking closely at a tiny 221 by 100 pixel grayscale image will be the tool that finds the exact mutation that dictates your medical future.
Your biological fate, the diagnosis that changes your life, could be decoded by an algorithm that learned to visually see patterns in the chemistry that human engineers couldn't mathematically define.
But I want to leave you with a final provocative thought to mull over. Everything we discussed today, DeepVariant, the custom color channels, the dynamic programming, haplotagging, it all currently relies on human-engineered hardware and human-designed data structures. Human engineers designed the laser sequencers, and human programmers decided to encode the DNA into a 221-pixel tensor. But if an artificial neural network can learn to see genomic errors better than our absolute best mathematical models, how long until the AI tells us that our sequencing hardware is looking at the wrong things entirely?
It's a thrilling, slightly terrifying prospect. Will the next generation of DNA sequencing machines be designed not by human physicists and chemists, but by the neural networks themselves? Will the AI optimize the physical chemistry, the lasers, and the voltage drops to produce the absolute perfect image for itself?
It is entirely possible. The AI might eventually figure out that we don't need a 221-pixel window at all, or that we should be measuring completely different quantum chemical signals that human biologists haven't even thought to look for. Because the ultimate goal remains unchanged since the beginning. That massive skyscraper of books is still out there, stretching into the clouds. The ink is still smudged, the pages are torn, and the text is vast. But we no longer have to climb it ourselves, reading every single letter by hand with a magnifying glass. We've built a machine that can step back, look at the entire tower, recognize the visual pattern of the typo, and point right to it.
We just have to be prepared for what it finds when the vision gets perfectly clear.